Hi, everyone. My name's Sebastian. I'm the host of Tears and Rain. And on episode 5, we have Danielle Beckman. And without further ado, tell us your story.
Okay, it's a lot. Like tell my story, like before I started my PhD, like
Y-
what got me in science.
yeah. What got you into science? How'd you decide to get to grad school? And then what were the key pivotal events that led to your decision to take on the challenge of getting a PhD?
Okay. Um, First of all, I think like I kind of can summarize my story with one sentence. Like I'm really a product of mentorship, good having good mentors. It's like because I was not a traditional kid that wanted to be a scientist. I liked scientists, I enjoy scientists, but I wanted to study history. I like writing. It was never like my thing, science. And actually, I went to my undergrad is in pharmacy, and I wanted to be a pharmacist later. I mean, I wanted to. It's kind of complicated, but I ended up doing pharmacy because I did my high school with a technical degree in biotechnology that it's supposed to be writing. But anyway, I started learning how to enjoy biology, but I wanted to be a pharmacist. I did not want to be like a scientist in a lab. And then like during my undergrad, I had like a very, very important professor that just gave this neuroscience class. And I still remember about it because my mind blows like, wow, this is so cool. Like what? Like this is what scientists are doing. And again, I'm from Brazil. So like being a scientist in Brazil, it's really hard. I mean, like in all the countries that don't have a lot of funding, right? So I was not particularly excited about being a scientist, but I got this professor and it was like I was totally obsessed with, okay, this is amazing. I wanted to work with him. I asked for him to do like to start working in his lab. He didn't have a place. I mean, he had no positions for me. So he suggested me another lab. And then I started doing my undergrad to do neuroscience, like being in a science lab and doing neuroscience. But still wasn't convinced. I wanted to do PhD. I was like, yeah, I don't want to be like, I'm going to be poor forever. And I mean, I already accepted this fate, but I was like, I don't know, being a scientist is too hard. So I was still kind of like deciding. Before my PhD, I did a master degree in biophysics. And because it was not that I wanted, but it was kind of like required in Brazil. If you wanted to do a PhD, at the time, you had to have a master before. But even during my master, I was still not decided I wanted to be a scientist. So I was doing clinical pharmacy, my degree in clinical pharmacy, and doing a master in biophysics, completely different topics. Um, but then, I was like, okay, like I really, really like neuroscience, and I was like, okay, I think I accept the fate that I'm gonna be a scientist. So it was not like I wasn't 100% sure in any moments, to be honest. And yeah, it took forever. I think during my postdoc, I was kind of like accept the fate. Okay, I am a scientist, and that's what I'm gonna do for the rest of my life. So I know this is kind of like trying to summarize the, the, the mess that it was.
It's okay. Everyone's life is a mess for sure. So was there any did you know the topic you wanted to get into when you were searching for your PhD program, or how did that work out? How did you decide on choosing?
That's a good question too, because so the thing is that I knew like I was very, very attracted for neuroscience. I wanted to study brains. But initially, so in my master, I was doing very basic science. I was actually studying prion proteins, like the protein that cause mad cow disease.
Mhm.
And and it was like I was in a lab that actually was not studying the bad form of the protein. I was studying physiological role of the protein, which I think is really cool because people talk about prion protein and they only think about the mad cow disease. They don't realize that actually a protein exists without being bad first. So there is a function for the protein, and nobody looks for the normal function of the protein. Everybody just want to see the disease. So I was in this lab like, okay, what's this protein is actually really doing? Like there is a function that is not only going bad, right? So, but I didn't like, I did not choose this. Like was a good mentor, like in my master that kind of like got me this topic. And I was interested about it, but I realized I didn't want to do my PhD on that because I wanted to do my PhD in something that I thought it was more related to humans and human disease. And it was the same time that my grandmother started to develop Alzheimer's disease. And I was like, like I had this training in clinical pharmacy, and I really enjoy diseases and trying to understand human diseases. And I was doing very basic neuroscience with prions. And I was like looking at my grandmother with Alzheimer's. So it's kind of like, I want to try to understand Alzheimer's too, you know? So like, it was something that, so it kind of like shift for me, like, okay, I want to actually do more translational science.
Gotcha.
Not that it's something, not there is a wrong or right. It was just something that got more interest at the time. Yeah.
Did your program have like rotations in the first year to try out different labs?
So that's a good point, because the thing is that the PhD in Brazil is very different, I think, than a PhD here in US. So at least, I don't know if it changed now. I don't think it did. But when I entered the PhD program, I had already to decide which lab I was going to work with. And I actually had to present, in the selection, I had to present my project. So yeah, so there was no rotation. Like all the 4 years I did in the same lab. And it was kind of like, of course, the project that I presented was definitely not what I was doing 4 years later, for sure. I think it happens to everyone, like things happen and change. But there was no rotation, so I was already fixed in this lab. But, and that's the thing, it was an interesting lab. And because it was very big, so there was a lot of competition. It was like 9, 10 PhD students at the same time, 7, 8 postdocs, like 70 people in the lab. Wow. So I kind of like find a way to go to another lab and actually be in peace. So I wasn't not. So that's what I mentioned about having good mentors. Like, I think I found someone that was my mentor that was not my supervisor, and I was with this professor following him everywhere and being in his lab and actually doing my research in this lab, even though I belonged to another lab.
Oh wow! How was the politics around that? Is it an allowable practice, or how did you get that position to happen?
It was more informal. So the thing is that, and that's what I always tell like people, like find very good mentors. Not necessarily, most of them are not going to be a supervisor, but try to recognize people you want to be around, and that it's kind of like can inspire you and teach you. So there is this old professor. He was like close to retiring. He was like he has this large space, but he was like knew a lot of neurochemistry, which was what I wanted to learn. And because he was older, he was kind of like respected by all the other professors in the department and everything. And he was not even accepting students anymore because he was already like getting ready to retire. But like one of my supervisor, my PhD supervisor, admired him a lot. So it's like, yeah, okay. I mean, yeah, you can stay there. And it's fine. I don't think he understood fully that I was full time in another lab. Because there's a thing, like my supervisor, my PhD supervisor had this huge lab, and he was also all the time very busy. So he didn't even realize where I was, to be honest. So I was kind of like doing whatever I want. But I think that's one thing. Like for me, I always worked like having mentors, supervisors that were like these super busy people that much older. That's not going to give you a lot of hands-on in the bench, and that doesn't work for a lot of people. I know a lot of people that prefer having a mentor or a supervisor that are going to be in the bench with them, are going to teach them directly. I wanted more the freedom. Like I enjoy working by myself and just coming with the reports. So there's like this balance, like what works best for you, right?
Yeah. For people who are watching that may not have the same level of independence or even confidence to do that type of work, I guess, just as a brief example, how did you come to that confidence? Like where did that form? How did you feel comfortable being independent and just doing the bench work and then giving reports? Yeah.
So like I was not just because I was independent from my supervisor, but not necessarily independent doing everything by myself. I was talking and getting a lot of support from other students, other postdocs. So I had like, it's not like I was isolated. It was just like, I prefer this mentor that there's a lot of mentors that want to, supervisors that want to have weekly updates and want to be closer. So I was like, okay, I want to do it by myself for the supervisor, but not necessarily I was alone. I was like bothering everybody around me and like, please help me. How do I do that? So I got a lot of support. It was just like, I was just filtering what I was letting my supervisor know, to be honest.
Cool. So could you tell us a little bit about the topic of your project and just like briefly summarize the pitch that you gave that secured the approval?
Yes. So what I did in my PhD, which is already completely different than I do now, it's I was trying to understand the non-cognitive symptoms of Alzheimer. So I was very interested in like depression, social isolation, anxiety, how this type of symptom can predispose people to have Alzheimer later in life. So for this, like, and I knew my passion was in neurochemistry, so I was like studying serotonin and dopamine receptors and how they can function or dysfunction if you have proteins that are seen in Alzheimer brain, like amyloid and tau. So this was like, but one very important thing, I never published my PhD thesis.
You never did. Interesting.
Never did. Yeah.
Oh no. So sorry to the viewers at home. There won't be a link down below for her thesis. Yeah.
Oh, exactly. I never published. And that's another thing, like we have so much pressure all the time, like you have to publish. And I left when I was like close to my finishing my PhD. I was like, I'm so tired of this lab and what I'm doing. I want to do something else. So like I left the lab, and my supervisor was not happy that I immediately left the lab after defending. So it kind of like my project was kind of like going through the drawer, and then later on it gave to another person, but this person never really continued the work. So I never published my PhD. And you can still be fine. You can still be a scientist and successful even if you don't publish your PhD. So
is there like a copy of the thesis anywhere that we could get our hands on?
In Portuguese.
In Portuguese.
That's the thing. Yeah.
Okay.
I didn't, that's embarrassing. But like at the time, I think they now do in English, but at the time, like they just, it was in Portuguese. So I had to put like even the figures and everything I had to translate and make in Portuguese because those they required at the university.
Oh, interesting.
So yeah, I do like publish a review or like these side things, but never really the title.
Gotcha. Okay, so that means you're gonna have the only oral history of the work you've done. So if that's the case, then could you walk us through like the years that you spent a little bit? Yeah, like the first year once you started and you're ready to go, how was that like in Brazil?
So it was very slow because one thing that most people don't realize is how like we are slowed down because things take forever to arrive in Brazil. So like if you buy any reagent, any antibody, it's just you have to plan like 3, 6, 9 months ahead, and sometimes sometimes even like that. It was so often like we're gonna receive, oh, your antibody is in the airport, and then you go to the airport, pick it up, and it's not even in the fridge. Someone just left in a room temperature somewhere. So this happens so much. So kind of like you have to be organized and try to like plan ahead and try to see like, okay, what I wanted to be studying 9 months and 6 months. So
wow.
Yeah.
Like, and do they give you a little bit of training for those contingencies? Like how, if you're a student, that could be panic inducing of just like, okay,
Yes.
you're going to start 9 months from now. Yeah. Like, how did you organize around that? Yeah.
I think that's a good thing that we learned doing a PhD in a country like Brazil is like you have to learn how to improvise. So like open your mind is like, okay, you have this major question you want to pursue, but what other side questions you have? What we have in the lab here that we can actually use to try to answer some of the questions you have. So it's always like having a plan B, a plan C, a plan D, and interacting a lot with others and just seeing like, because I think sometimes here in the US, the labs and the departments are kind of like isolated. Like each lab don't communicate so much with the others. But when you are doing a place like Brazil, like you have to know exactly what the other lab has, like what reagents you can use. And like we share a lot because that's the only way you can do science. You have to be sharing. Otherwise, everybody's going to be isolated waiting for the reagents forever. So, yeah.
Wow. Yeah. Did you, so how many, if you can roughly estimate, how many plan A, B, C's, and D's did you have going into this?
Several.
Yeah.
Yeah, it was. Yeah, because I remember like when I first started, I really wanted to study serotonin and look at the serotonin receptors. And we kind of like, we did not have the best animal models for Alzheimer because they're very expensive. I want to like have the super transgenic mice and everything, but we can't. So it's just like, it's very expensive. Jackson Lab doesn't ship to Brazil or whatever. There was always a complication. So it was like, okay, how can we study? I tried to, like, can we induce, so we did not have the mice that actually had the amyloid pathology for Alzheimer that I wanted. So we're kind of like starting injecting amyloid in the brain of the mice, kind of like trying to induce what the transgenic is born overexpressing the protein. We were just injecting the protein in the brain.
Wow. And does the, sorry to get a little technical, but does the protein diffuse into the tissues? Wow.
Because we were injecting in the ventricle, lateral ventricle of the brain, so trying to make it diffuse.
Yeah. Wow. I'm biased here. I love like scrappy science, like trying to figure out how to do really good science on a minimal budget. And so was this like common practice, the injection direct?
Wow. Yeah. So we ended up, that was something that was working well for the lab. So we kind of like created our own version of mice with Alzheimer. And then the whole lab started to work with that. And because some people were doing behavior, so we're kind of like seeing after we inject the amyloid, and we injected freehand in the mice brain. It was kind of like wild. And the mice was developing memory loss, like some of the things we see with Alzheimer. It was just like our cheap version of the fancy Jackson Lab transgenic animal, and it was working. So ended up that a lot of people in the lab was doing is just looking at different proteins, actually, but using the same auto.
Wow. Yeah. So you essentially like built a tool that was useful to like the people around you. That's very cool community science. Okay, so the first year was slow. Then the second year, now that things are working, did things start picking up, or?
So the second year, I think, was kind of like when I realized that I wanted to travel more. So I always enjoy traveling, and I wanted to travel more, because every time I went to a conference or a scientific meeting, whatever, like I saw that I was learning so much. So it was like I spent a lot of, I'm not ashamed to say that, I spent a lot of my PhD traveling for scientific meetings.
Cool.
And I think an advice I can give to people is like, people that are starting their PhDs or thinking about starting their PhDs, like get involved with the societies, scientific societies in your field. Because like, I remember when I was studying and I wanted to travel more, there was like in neuroscience, the biggest society for neuroscience. And it was like, people, oh yeah, just go to the meeting. But one meeting was just once per year, and it was in the US, and it was super expensive. But then you start seeing like, okay, there are these smaller societies that nobody talks about. Like in Brazil, there was the Brazilian Cell Biology Society, the International Society of Neurochemistry. And then like when you start digging into the websites and about the societies, they have a lot of fellowships for people that are outside US. So I always travel like getting these awards for Latin American students. So like they do have these things. And I remember talking to people like, why are people not applying? People just don't talk about it, or just don't dig enough about these societies. And like, you can really learn and actually get a lot of travel fellowships and awards just if you can keep applying for everything. So I was just applying for everything and trying to get like the obscure little societies that nobody talk about, but it kind of like have a little funding. It was not a lot, like $300, $400 here, $300 here, but just like get involved and try to see, because the more you apply, the more you're going to get.
Sure.
This awards. So I think during the second year, like everything was super slow still. I think things, because I did my PhD in 4 years, but it started, but I had the master before, which was already 3 years. But I think during my PhD, like the first 2 years, I wasn't doing much. Even like, I think basically all the results were like a year and a half after, like in the end, like it's usually how it is, right? In the third year and the fourth year, okay, finally things are starting to work.
Yeah, all the previous.
I was traveling a lot.
Yeah, all the previous speakers were saying roughly the same. That is just like things weren't working and things didn't work, and then at the very end things worked, right? So it's like an exponential progress. So what was like the most formative experience during that travel year when you went to these smaller conferences? Like what did you take away, and how did that shape you going forward?
Completely. Like first of all, I met my mentor, my current mentor, the one that make me move to US, in one of the schools. It was a super small school, like 6 or 7 students. And he was one of the speakers, and I never heard about him before. And he was like, oh, yeah, I just like mind blown about, he worked with monkeys, and I was like, oh my God, this is so cool. He's trying to make a monkey model of Alzheimer. because monkeys don't have Alzheimer. Only humans have Alzheimer. And why monkeys don't have Alzheimer, even if their brains are very developed? So like, can we make them try to induce Alzheimer in the monkey brain? And he was talking with me, and I was like, wow, this is so cool. So I think the most important thing is like, I found my current boss and mentor in one of these schools. And also like, I just learned about different types of science and different ways of doing science that I wasn't familiar with. So, so I think it completely changed like everything, to be honest. And just connecting with people and just meeting people from different countries. And it's just like, I remember like visiting labs, like I visited a lab in Japan, I visited a lab in China. It's like, damn, all the labs kind of like look the same. We're all the same, we're doing the same type of stuff, and we're interested in the same things. And it's just like, we just need to connect more and talk more with people.
Yeah, that's a really good bit of advice, especially for PIs, to just kind of reach out a little bit. Because I always hear, especially across fields, that there are like parallel processes that are happening that would really benefit from communication across fields, right? Especially like biochemists and biologists, or ecologists and biochemists, things like that, right? Like these type of like frustrations that I hear, especially from clients. Like I make my bread and butter because there's not that much communication between fields. Like I do cloning for labs that have never cloned before, for example. So within those labs, you said that everybody's kind of doing the same thing. Did you feel, even in the slightly more well-funded labs, the same type of spirit of exploration was the same, or did you feel like there's an inverse relationship between funding and passion?
Yeah, there was. Yeah, I mean, it's bad to say that, but because being fairly honest, most of like, when I visit the labs, I did not like visit any lab. I mean, yeah, I visited one lab in New York, like in the middle of my PhD, but most of the labs I was visiting was like, so I spent like 3 months in Chile. I spent like 2 weeks in Japan, which was not a well-funded lab, even though it was in Japan. So I was more spending my time in labs that were like more similar to my reality. And when I saw the labs in US, or I didn't even see any in Europe at the time, I was like, okay, wow, there's a lot of these pre-made kits that you mix A with B and then add C, and people have no idea what is A, what is B. It's just like everything is like, there's not a lot of troubleshooting, I think. It's just like, oh, okay, we can buy this reagent. And it's just, I mean, this is the reality of my reality when I moved here to US. I saw like the science that I'm doing in the lab now, it's very different than the science I used to do in Brazil too. And that's fine too. It's just like, you have to keep the curiosity, but I don't need to, I'm not required anymore to have a plan B, a plan C, a plan D. And this is, it's good, but it's not too, I think, right?
Yeah.
It's,
yeah, it could really, like, I always found interesting the components of the kits, right, and what they were on. And the kits, so like the well-made kits in the manual, they have references to papers that they was based off of. And reading those was so informative, especially like I have no formal training. So like my training was those methods papers, right? And then like starting to understand like why is there sodium hydroxide in the lysis buffer for mini preps?
What it's doing.
Yeah. And it's extremely helpful. And I've seen and haven't seen at the same time the level of depth of explanation of some of the processes, and rather just like, oh, doing a mini prep requires a kit, right? And then so like when I published my protocol on my notebook, I had to call it a columnless kit, because the kit, like a columnless mini prep, because the columned mini prep is the de facto mini prep now. Like people don't necessarily are familiar with like ethanol precipitation, right? Which is a little frustrating because it's super handy and sometimes you want all the DNA, for example. But yeah, like my bias is threadbare. I'm a big fan of the resourceful, scrappy kind of do-it-yourself style. Okay, so the traveling invigorated you, gave you your mentor, and now let's talk about that 1.5 years that you had to like blitz your project and like floor it.
Yeah.
Yeah. So how was that? Yeah.
It was pretty wild, because like when finally the things were starting working, so we have like, okay, we have an alternative Alzheimer model. Like I learned how to inject the protein. We're seeing differences. So it's just like, okay, I need data because I don't have enough data to defend my PhD. So I was basically freaking out. just driving around the city trying to get where I can buy mice, because it was like this. It happens sometimes. There is no mice available. What am I gonna do? I was doing. It's very different because I work with monkeys now, but it was like doing 40, 50, 60 mice a day, injecting them, and I was doing a lot of behavior, and that's. And my supervisor had no idea I was having all this problem, but I had all these friends and like this community that was helping me. It was like, okay, she needs data because she needs to defend her PhD. So like all my friends are like, okay, like helping me with behavioral analysis and like trying to help me with the microscopy. One of the dull things about being in a country that is not very well funded is like, you cannot, you don't have permission to break equipments, or like you cannot commit errors, otherwise they're going to be very expensive. So I spent my whole PhD avoiding the microscope. I never used I am a microscopy nowadays, but like during my PhD, I was so afraid of touching the microscope and breaking it. So I spent my whole PhD avoiding microscopes. And then like when I moved to my postdoc, I was like, oh yeah, okay, I can try this. It's like, oh my God, this is amazing. Okay, I want to do that for the rest of my life. So it's just like I had friends that were doing microscopy for me, and it was just like this community of other people and other scientists, and from different levels. Undergrads were helping me a lot. I just had a lot of people like, okay, she needs data. She has no data, and things are getting bad. So yeah, it was pretty stressful. The last 1.5 years, it's funny because I have so many memories from the beginning, but the last 1.5 years, it's almost like I blocked these memories. I'm trying to retrieve now. And I just like, I'm starting to sweat. I just feeling the pain. It's just like, it's so traumatizing. It's very stressful.
Yeah, I deeply appreciate the vulnerability. And I know digging this up is heavy, but for students listening, I think this will be really beneficial, especially for international students who don't have that luxury of having all those resources. Is that sense of community something that you found when in your current position, like that sense of help and belonging, or
never the same? It was I never had the same. I mean, I don't know if to be fair, it's like I'm kind of like in a very isolated place because I work at the primate center, so I don't have. It's not like I'm in the middle of UC Davis, right? I'm in the University of California, Davis. I'm not in the middle of the campus. I don't see undergrads. I'm just isolated outside of the primate center. So to all fairness, like I'm in a very isolated place, in an isolated position. But even inside the lab, I never saw, I think people here, I don't know if can be my own experience, but here people work much more independently. Like when I see other people in my lab, other postdocs, other researchers, are just like, they're doing their own things. And in Brazil, I felt like it was just like, we have to, it's a chaos, it's a mess, everybody freaking out. So we kind of like have to give the hands and like, okay, let's do this together. And we can, so I never had the same sense of community I had during my PhD here in the US. Yeah.
Yeah. Wow. The, so in your fourth year, you're about to defend you put the last period on your thesis. How did that feel like? Like, one of the questions I like asking is like, did you feel like you were an expert in the topic you were addressing? No,
of course not. Yeah. No, absolutely not. I was just like, do this even make sense? I'm like, I wrote all these pages is like, okay, I know a lot about this very specific topic, but what does that mean? It's like, no, I did not feel any at all. I think like the only thing I could feel was relief, to be honest. It's like, because it's such a painful process to write. I think we don't talk too much about how painful it is writing your PhD. And there's so much pressure when you start, which part of the text you want to start. A lot of people know. I remember getting so many people advising me like, oh, start with the results, start with the material methods, I wanted to start with the introduction. I want to start in on how we read. And I remember my supervisor like, okay, you have to send me the chapters, and I did not have anything yet. It's painful. The writing process is painful. But so I think like I was just so stressed out that when I ended, and even after defending it, I was just relieved. That was the only thing I did. But I did not feel, and I still did not feel at all any specialist or anything at all. Nah, no.
So you submitted, you defended, did the, just briefly, like how was the defense in Brazil compared to the States from what you know?
I think the defense is kind of like similar. I mean, I think here you'll have like an hour or something to talk, and then you have like there's some faculty that is going to judge you, right? So it's, I mean, not judge you, but it kind of like ask you questions of what you're doing.
Grill you. Yeah.
Yeah, judge, right, basically, right. Yeah, judging me. And it was a lot of pressure for me because I like have two of the faculty that came from my PhD defense was from different states in Brazil. So I went to the airport to pick them. It's always have to be yourself. Like, it's like, okay, I went to a hotel to pick one. Another friend went to pick the other one in the airport. And it's like, there was a problem with the hotel. And I'm like, I have to defend my thesis. And I'm like paying the bill of the professor in the hotel. It was just a chaos.
Oh,
yeah.
Wow.
So it was just like, so it was stressful, but I think it worked out. I mean, yeah, as most people, it's just like, I think it's more like you controlling your own emotions than because if you're in the point that you're like giving a presentation, people already read your thesis. I mean, it's very hard to screw it up, right? And like I've seen before, but it's rare. It's rare to screw it up. Yeah.
So you said that the work you did for your thesis doesn't necessarily match with what you're doing today. So could you first tell us what you're doing today, and then we'll talk a little bit about how your PhD experience helped that.
Yeah, and again, a product of having good mentors is just like it completely changed everything. So it was like, yeah, I was doing Alzheimer research, and I have my supervisor now, which is my mentor. It was just like I was studying Alzheimer. We're trying to make monkeys have Alzheimer, and then he was the director of the primate center, and he was like, okay, we should not waste brains because monkeys are precious, and there's different projects in the primate center, and people are not looking at the brains from different projects. So he was like pick the brain for us, and he was basically collecting brains from different monkeys from different projects at the primate center. So if you go to our fridge in the lab, you're just gonna see a brain of different diseases or different things that people are studying, and one of them was SARS-CoV. So COVID-2, COVID-19, yeah, exactly. So it's just like there was these brains with COVID in the fridge, and it was like, okay, let's look at that. I didn't know it was COVID. I thought it was Alzheimer at first, and then like I started to look at viruses in the brain, and this is what I do now mostly. And then I started looking at HIV and Zika virus. It's like, oh my God, why these viruses are entering the brain? So now I'm like working less and less with Alzheimer and more looking at what viruses are doing in the brain. And I think like, and my mentor, he embraced that. He was like, okay, this is not the line of research that I am interested in. My lab is in Alzheimer, but I'm okay with you doing virology because I see this as important for you.
Wow.
So
that's huge.
Yeah,
that's really huge.
It is huge. It is huge. It is huge. Like having a mentor that allows you to find your own line of research and exploring that and supporting financially and everything. Because I completely like, okay, we have to have another postdoc in the lab to replace you because you're not doing your Alzheimer research as you should. But you're doing something. He believed in it, and he saw that it was important for me. So.
Wow. And with that support, does that come with like room to publish where he is the PI leading that project, or because it's kind of distant from his expertise, right?
Yeah. Yes. I mean, he got excited of like, I mean, he's the same style. I always have the same style of the PI, which is like the senior researcher. My supervisor is like 75 now. He's probably going to retire in a few years. So he's just like more relaxed. I think, I don't know if he would behave the same way 20, 30 years ago, but he's like, okay, like this is interesting. I mean, and there is some connection. Like we think that the viruses in the brain can cause some neurodegeneration similar to what we see with Alzheimer. So yeah, like he has been an author for the papers, and yeah, I think he's more open to the exploring this.
That's awesome. That's really, really interesting. Okay, so could you dive a little bit more into the specifics of the work you're doing now? Like what's a part of your project that's really, really interesting for you? Yeah. If you can.
Everything. No, everything. I'm fascinated by microscopy, and if I could, I would just live in the microscope because It's just like this feeling like I remember like the first time I saw COVID, SARS-CoV inside the brain, inside a neuron, my mind, I remember exactly what I was doing, what I was wearing, because it just blew my mind, like just this feeling like, wow, I'm probably one of the first people in the world seeing this.
Wow.
And this is, yeah, is this feeling that, and again, like for someone that was completely avoiding microscopy during my PhD, I was so scared of microscopes because they're expensive, they're fancy, I'm going to break this shit, it's going to be horrible. And it's actually no, and another advice I want to give is like you have to try really hard if you want to break an equipment in the lab. Like you have to try really hard if you want to break a microscope. It's not that easy to break, so try, and people explore. I regret not exploring my PhD because I could have had so much more fun because I love it. So I could be just having way more fun way before instead of having now. So it's just like be less like worried and obsessed about not breaking things and really explore and try, because it's really hard to break it. And so I just spent a lot of time in the microscope taking photo of weird things in the brain.
Yeah, I mean your photos are gorgeous. And did you do you have a place that you store them for public viewing? Like, do you have a gallery of sorts? Because I always try to advise people to definitely do it because it's beautiful. It's art. It's living art. Yeah.
That's something I want to do. I'm just trying to find a way that I can do it that makes sense, because one of the things that happens a lot too is that people stealing your images, like people just don't get the credit or just like copy your images and publish somewhere else. And that really is not cool and it's very stressful. So I was like, okay, how do I deal with people just stealing my images? But at the same time, I want people to see them, and so I still don't have, like, I put some in my website, but I have to put more. So I have some of them are in my website. Okay. Some of them are in my website, but I post a lot on Twitter and Blue Sky, but I don't have yet something organized that I can actually store them, which is a shame. I have to find a good way to do that.
Yeah. The episode with Minya, she has a lot of electron microscopy of flower meristems. So maybe contact her with like how she came about with her design and her feelings on like misappropriation of her work, right? Because that's another worry, because you have that adjacency between SARS-CoV research, virology, and then images that people could say are false and things like that. Because it depends if you want to even touch that, but about your feelings on that, please tell me. Yeah, like I know that you've been crusading pretty strong against a lot of the misinformation online regarding this from a really empirical stance, not a trust the science, but here is the science, like here is the data of it. Yeah, so tell us a little bit about that if you can.
I feel like what's happening is just trying to, so I was just posting about this on Twitter, like I'm just a shy microscopy, I just wanted to do my science in peace. But like I could not live with the fact that I was seeing something major and seeing people not understanding or not even have any idea that this is happening inside the brain, and like this type of thing exists. Like even nowadays, if I post a photo of COVID in the brain, there's someone saying that is not true. You cannot see viruses with a microscope. Like people keep saying that. Oh, you need an electron microscope. So people don't even follow the technology, doesn't even know what is high resolution microscopy is, and just like. And with one image, you can convey so much information, right? It's just so easy and more accessible than just, I think I was growing tired of just seeing like how complicated the scientific manuscripts are and how inaccessible they are for most people. And like, okay, how can we pick whatever, there's a very, very important information in this manuscript, but how can we translate that people can understand what's happening and where science is going? So I started to just share about this online. And it's getting a lot of attention, a lot of hate too, a lot of people that don't believe in science. So it's pretty stressful, but I think it's necessary. And I think we kind of like, I really feel like we are scientists, and I'm paid with NIH money, right? So it's like the public is paying my salary. So it's like, I feel like it's kind of like our duty, especially if you have something like pandemics, viral infection, or something that is like, it's going to, long COVID is making a lot of people sick. It's our responsibility as scientists, like tell the public what is happening and tell our point of view. And each person can decide if they believe or not, but at least we are paid by the public, so it's kind of like our duty to communicate about the findings, I think.
Yeah, I appreciate that stance so much, because there's an interesting divide in critical thinking when it comes to these things. And I mean, we can't expect everyone to be a trained neurobiologist in order for them to make a decision that it can impact their lives, right? On things like COVID, on how the pandemic was done, how safe the vaccines are, things like that. But I mean, your images were so compelling. When I remember messaging you through a DM, going to, I can't believe those are COVID spikes and that they're inside, right? Like the
how is this inside a neuron?
Yeah, like passing the blood-brain barrier, sure, but like inside of neurons. Do they replicate in the neurons as well? Wow, that's terrifying. So
because it's like, because the neurons communicate so fast with each other, right? So basically the virus uses the same communication system that the neurons are connecting. The virus hijacks that, so virus can actually spread much faster in the brain just because it's using the neuronal connections.
So they like spread through the synaptic cleft from one place to another. Wow, and are there sorry to get super technical. This is so fascinating.
No, I can speak about that for 10 hours. Great.
Yeah, like what in between the synapses, what on the surface is the virus attaching to, or is it just going through vesicle from one to another?
Oh, I mean, this is so, I don't have this type of resolution. This is more EM, so I don't see direct. What I see at my level, I see the neurons infected. I don't see myself, the neuron, spreading the virus to another, but there is other groups doing that, and they see by vesicles. So it's like, I mean, the neurons have a lot of ACE2 receptor too, which is the receptor we know that the spike protein from the virus binds. But we think mostly it's spreading through vesicles, from neuron to another.
For folks who are looking at any of the images that you might be willing to share, and there are fluorescently tagged proteins. Are you tagging the proteins? Are you tagging the virus directly somehow? How are you getting the color differentiation?
So, great question. It's the proteins mostly. So as soon as the SARS-CoV pandemic started, there was these companies that developed these very good antibodies. I mean, the beginning was a bit hard because the antibodies were not that great, but it's basically, I'm targeting different proteins from the virus. So like a lot of people use spike protein, which is like very specific for SARS-CoV-2, but we also have, because you're doing, I'm doing multi-fluorescence microscopy, I can use like different markers for virus, or not only, because sometimes if I only post spike protein, someone's going to say, oh, but the vaccine is causing that. Which is fair. I mean, spike protein is in the vaccine, but I can also see double strain RNA inside the neuron. I can also see nucleocapsid protein from the virus. So what I try to do is like trying to use different proteins to target the virus and combine them with proteins like proteins that are only expressed from neurons, proteins that are only expressed by microglia. So kind of like combine them and see where the virus are and what the virus is causing. But it's mostly the proteins,
And with the capsid proteins, do you get the same level of fidelity as you would the spike proteins when it comes to tagging?
Yeah. It does.
Wow. So that's what I love about your images is that it doesn't require much trust, in sense of like, you don't need to have like, oh, clearly she wouldn't lie or anything like that. It's more of just like, here are these images that with good controls where the physical protein is being trafficked neuron to neuron, right?
Yeah, exactly.
Yeah. Wow.
And I think one of the things that it's most impressive for me, and I think for a lot of people, and you can see clearly in my images, is the inflammatory response, right? Because it's quite clear. There is still a lot of debate, even in a lot in neuroscience field. There's still neuroscientists that don't believe the virus enters the brain, even though I have images and other people have images, people still don't believe. But a lot of them realize that the inflammation is real. So there is a lot of inflammatory response in the brain. And I think this makes the visual impact of the images very strong, because each inflammatory process is going to look different to another. So that's the thing, like the normal brain, it's, let's say a crime here, but the normal brain is boring in terms of like
spicy.
Yeah, because everything is pretty, and the brain is so well organized. So you see everything like the cortical layers, and everything's well structured, and the microglia is not even touching the neuron so much; it's just keeping the distance. Microglia are the immune cells in the brain, and when you see inflammation, and that's why I'm fascinated about microscopes. Like each inflammatory response is going to look different. So I see this microglia attacking this neuron here, and then I move a little bit to the side, and I see another microglia attacking another neuron, and it's going to be completely different because inflammation is unpredictable, and the cells go crazy. So I think that the visual impact of seeing inflammation in the brain when you have viral protein as well is what attracts a lot of attention to.
Is there your personal opinion, but is there any particular reason why some scientists don't believe that viruses are in the brain?
Because during my PhD, I also received the training. We received the training that the blood-brain barrier. Well, first of all, blood-brain barrier. What do you think about barrier? Like a wall or something that blocks, right? Mm-hmm. So we learn, and I learned that too during my PhD, that the brain is protected, the brain is isolated, the brain does not allow other things to enter, is is highly selective, and there is no way the brain is going to let these viruses or bacteria or whatever enter. So we learn, you basically have to give up your learning and relearn, and a lot of people are not willing to do that because, like, if you see the history, it's just like not a long time ago, scientists and people were saying, the scientists were saying, like, no, these things don't enter the brain. And then it's like, suddenly you like, okay, yeah, you have like very highly selective, but the barrier is just a bunch of cells together. There's layers of cells, and the virus learns how to hijack these cells. So, but this is so new, like, this is like very, like, it's getting bigger now with COVID. So I think a lot of people still believe that the brain, it's the blood-brain barrier, and that brain is immune protected is still a thing, even though it's not. Yeah, so it's just like relearning. Yeah.
Yeah, it reminds me of that "Life Finds a Way" quote from Jurassic Park, because, like, it's interacting with it. But do you think that there was like a recent change in access to technology or to certain technologies that allowed for us to explore this to the point where we can say that there are viruses that pass the blood-brain barrier?
Yes, but it's also, there is this challenge that, and it's a big problem with COVID, because, like, in the beginning, when the pandemic, when, like, people were looking at the brains, they were not finding the virus in the brain, because that's the thing, like, the brains are like, the people are like sitting in the piles, like, especially in New York, there's so many deaths. It's just like the brains were not. So if you wait too long, the virus is not going to be in the body anymore, right? Mm-hmm. After the person died, because the virus needs the cells. So this slowed down a lot of the research, because at the beginning we're not like having the good, what we call post-mortem period of time to process the samples. So if you're studying virology, first of all, you have to work with high resolution microscopy or electron microscopy, because they're really tiny, so it's really hard to see. So, and also, so a lot of labs don't have access to high resolution microscopy and stuff like that. But also the fact that you have to process the brain samples or whatever organs fast, otherwise you're not going to see anything. So there was a lot of papers, and there are still people citing them, saying that, oh, the virus doesn't enter the brain, the virus doesn't enter the kidney. It was just because, like, oh, and you're going to check how long it took for them to process. Oh, the brain was processed 3 days after the person died. So yeah, of course you're not going to see any virus. So there is like this challenges of the field, is like how we are getting better. The technology is definitely getting better, but it's happening right now, and it's slower than it should, I think.
Yeah.
Especially with bird flu now and all the other stuff coming.
Yeah, it really does sound like, not to drop another quote, but the whole absence of evidence is an evidence of absence thing, right? That's one of my favorite quotes, because I try to think about that a lot with my own experiments. I mean, nothing compared to the seriousness of what you're doing, but from a perspective of that, are you seeking to continue this line of research? Like, just to shift gears a little bit. So you're currently a postdoc and you're on the job market, right? Yeah. What would be your dream position given what you know? Would you want to continue this type of viral related research? Yeah.
Yes, and that's has been a big frustration, as I shared with you before. I want to do neurovirology. I want to have a neurovirology lab. And it was just being really, really hard to find a position, because I don't see interest from the neuroscience department. So I have been trying both, applying for neuroscience departments and for virology departments, and I'm being rejected from both. Neuroscience departments are not seeing like I think the importance of studying viruses in the brain. I don't know. And virology is like, oh, you should be applying for neuroscience. Like you're not really a virologist. So it's just being really, really hard. And now with the government changes in US and all the complication about doing infectious disease and all the anti-science movement, people are not believing in vaccines and all the stuff is just harder. So it's just like I'm looking for options outside US too and seeing. But yeah, I want to definitely, this is like I want to do for the rest of my life, is just look at these viruses in the brain. Yeah.
Wow. I mean, from the beginning, when I first saw your first images, it was like not only compelling, but like very good material for coursework for educators. Have you ever considered making like small modules for curricula to explore, like here's an evidence-based exploration of viruses in the brain, something like that?
No, that would be great. Like one of the things I dream about doing, and I taught a course like online before, but it was just like 1 hour. It was not really a course, just a small thing. But I wanted like show people and teach people how to do microscopy and how, like basic, like how to combine which markers you should be, if for the neuroscience, which markers you should be targeting, why you need to do everything in 3D. I hate so much when people are doing 2D microscopy. It makes my heart sad, so sad. Like people saying colocalizing proteins and doing 2D, just one single image makes me sad. How do you colocalize? So it's just like the importance of doing 3D microscopy and properly quantifying things in an unbiased way. So I want to do like so bad a course about this. And I've been trying like, okay, should I do a course in university? But maybe something online, like modules, as you suggested. It's probably a good idea.
Yeah, like there's one of my favorite course websites, Udemy, has the ability for you to make your own courses, and it's like online. You can give a little certificate at the end of completion. But
I think I never thought about this because like I'm doing a course, like do I have the knowledge necessary to do a course? Because all the time when we think like, oh, someone is doing a course, it's just like someone has a lot of knowledge about the field, but maybe you don't need to have, I don't know. Maybe I can do a model. Yeah, I don't know.
Yeah, I think for things, especially with your research output, the images can't be made without skill and understanding, right? You can't just like randomly make that, right? You like just do stuff with a microscope and these beautiful indicative images occur. So clearly you're skilled at that thing. And I think it would be important because you have a tacit knowledge, an immediate, fresh tacit knowledge, because you're in the trenches. You're like a living microscope at this point, right? Of being able to do those things, right? And I strongly recommend, if anything, just to compile those tips into something that's easily communicable. Yeah, I'm a huge fan of just like information exchange at whatever level, because as long as what you're doing is clearly evident that you're doing something in a manner that's positively productive, it's credentialism doesn't matter. Like you can do the thing, so you should try to do the thing and share that information. And if somebody gives you feedback of saying, that's not how you do it, you should do it like this, right? Then that's a moment for you to be able to grow as well, right? Because you can be like, oh, I never thought of that. Or, oh, actually, I've been doing this for X amount of years and this is my body of work, right? Which is why it's important to have that gallery, right?
That's a good point. Yeah. I think it's all about, and I mean, you probably know this well, like the scientists, we suffer so much from imposter syndrome, right? Like there's not a single day like, oh my God, what I'm doing, I'm a fraud. Like I don't know anything. It's just like, it's just this self-consciousness that is like, I'm not good enough. I'm like, why am I going to be teaching people if I don't even know if I'm doing right things, you know? It's just like,
yeah. As the patron saint of imposter syndrome myself, I like not having a degree, having interactions with scientists. It's always imposter syndrome, right? You always feel like a fraud. But like being an uncertified fraud is like a unique, different kind of fraud, right? But at the end of the day, like you said, with your explorations of other laboratories, is that we're all human. We're all doing the same thing, right? And if no one has the answer for how to fix imposter syndrome, but I'm hoping that stories like yours could just show that even though you felt like you didn't belong the entire time, you still got to where you got, and you still have a respected body of work, and you're continuing to do these type of things, right?
That's true. And also one thing like I want to mention, like for people listening, that is just like imposter syndrome for me, like, and for many of people that don't have English as a native language, like, am I speaking things right? Am I making sense? Can people understand me? And it's just like, I always, even like I have been living in the US for many years now, but I still have this thing like, am I English good enough? Because you always have this feeling like, okay, I sound so much better in my native language. Like I could be so much more cool. And then at the same time, it's like, oh, my accent is too bad. Like, can people understand me? But then I think about, and someone told me this, and I kept this with me. It's like Americans, like, and you can tell this more. Like Americans are so used to different accents, right? It's okay. It's not a. 100%. It's okay to speak with an accent, right?
Yeah.
So because I see, like, I had this problem a lot myself. Like, okay, I feel shy about talking because I don't think my English is good enough or my accent is too strong. And the reality is that Americans are not even paying attention so much because you guys are so used to accents all the time. So I think like one thing, if I could go back and tell myself,
I was going to ask you that.
yeah, be less shy and more open, and because there were so many times I wanted to say things, but I was like so self-conscious, like, oh, I'm going to sound weird, or like people are going to understand me, or it's just like, they are going to understand you, you know. It's just like, it is okay. And people want to hear your message, not how you're speaking, if you have an accent, whatever. So I feel like for young people, and if I could tell myself and anybody entering the PhD again, is that don't be shy about communicating and telling what you think and what you feel is important because of the language barrier, you know, because that's a big thing, I think, for non-native American or English speakers.
Yeah, there's a lot of folks from certain countries that feel like if you don't speak their language properly, it's a huge insult. It's
true.
Yeah. I'm not going to name any names, but
France.
Yes, definitely. But from the perspective of like doing science in America, like even in my undergrad, every single teacher was from a different country and had a different flavor of English, or different color of English that they spoke with, and that really doesn't matter, right? Like, in fact, it's one of the few places in the world where you can have all of these different places, and we're still trying to get to that same message, like you said. You know, like I remember there was a funny meme about Sofia Vergara, who's just like, I am so much smarter in Spanish than in English.
I use this all the time.
Yeah. And yes. And I mean, I'm hoping with technology that translators can be direct and whatnot, but there's another quote that I remember. I'm paraphrasing poorly, but he said something along the lines of, if someone speaks in a really heavy accent and you're a native English speaker and you don't understand them, then you have not mastered the English language.
Wow.
Right? Because your brain's not able to parse all the different nuances, which means that you didn't master the English language. Right?
That's a point.
And that really resonated with me, because if I hear somebody trying and they're trying to communicate and you give them patience instead of like correcting them on grammar, which I absolutely hate, like, especially verbal, like on written paper, I understand because it's a document, it's communicable, sure. But in language, it's such a vulnerable moment of insecurity, right? And it just, I'm a city kid. I grew up in New York City, right? And so it's like we're
used to all the language,
every color under the sun in terms of English accent, right? And no, it's
but that's such a good point, because I feel like when we start, people that are entering the PhD, like, you're already reading so many papers, so you know how to read English very well. You probably write well as well. It's just the speaking that takes so much longer than the rest. And I spent so many years feeling like a fraud, or not avoiding communicating with people and telling what I think, because I was like, okay, I write well, I read very well, but I'm not ready to speak. So I keep delaying. I'm not ready to speak. So like so many places I visit that I wish I talked more to people, I was just avoiding because I was afraid of being rejected. And the reality is what you said, like, if people are not understanding you, it's probably not your fault. It's probably they're not doing a great job learning their own language, I guess, right?
Yeah. And it's already not the greatest language to learn if you don't have the developed sound house for it, right? Like
Yeah.
certain people will carry their accent forever, no matter how long. And it's just, especially in the sciences, it's just the fact that we kind of have to understand, right? Like
Yeah.
if you,
It's
if,
a universal language for science, and it is. Yeah.
yeah,
There's nothing you can do about it. Yeah.
right, right. And it's, what's more important is, like you said, the message that you're saying. So with the minutes remaining, is there anything you want to plug or pitch or talk about? The floor is yours, wherever you want it to go. Please feel free.
I don't know. I think it's well, I think one of the things that I don't know if we covered, but it was just like this idea that doing a PhD is a big deal. And it doesn't matter if you're doing Brazil, in India, or in Spain, or whatever, like, it's a PhD, and you should be proud of that, and you should be confident about your knowledge. Because I remember when I first moved here to US, I moved right after finishing my PhD, like a few weeks later. And I was feeling so imposter syndrome again. It's like, oh, I'm not good enough. Like, I have all these other people in the lab that have their PhDs in US. And I was thinking with myself, oh, my PhD in Brazil is probably not as good as them. Like, I probably did not, I don't know why, but you have this like, oh, I'm coming from a not so developed country, my training was probably not as good. So this is such a lie. So I think that my message would be like, if you like did a PhD, you already worked hard enough, and you probably have a lot of the knowledge, even more, like, because you learn how to troubleshoot, how to have plan A, B, and C. So feel more confident about yourself and about your learning, about your knowledge, and less preoccupied and obsessed about, am I good enough? Should I be here? So I think my message is just like having more self-confidence. If I could tell myself, like, when I was starting, because I struggled a lot when I moved here, it's like thinking I'm not good enough. My training was not good enough. And that was so not true. My training was more than enough. I did not have any other more problems than any other people in the lab. If anything, I knew how to troubleshoot more. So it was actually a little better in a lot of aspects. So I feel like, don't feel discouraged or embarrassed or shy if you want to move to US or to Europe after you're doing your PhD in another country, because you're going to see, like, you know, probably way more than the people that were actually around there already. So feel confident and feel, confident about your knowledge and about, and less preoccupied of others are thinking, I feel. Yeah.
That's a good message to take home. Well, thank you so much for hopping on the call. I hope you enjoyed it as much as I have. I can't wait to talk to you about more of this stuff, especially after we stop recording, because I could ask you questions all day about all the neuro stuff. I'm a little bit of a neuro dork. But yes, thank you so much. And we hope to see you on again.
Thank you. Thanks for having me. That was great. Thank you.